Hi, Victoria. How are you? Hi, everyone. How are you? We'll start in a few minutes, waiting for the speaker to arrive and for more people to arrive. So, thanks for your patience.
Sounds good.
Katerina.
Welcome, Jane.
Hello. Hi, Victoria. How are you?
Very good, thank you. Just got home from a hike. It's really sunny out here. Kind of warm, but not really. How are you?
I'm fine. It's like 2 a.m. here. A little bit chilly, but I'm sorted. I've got a nice cup of tea to keep me warm through the presentation. What about you, Katerina? How's your day?
Good, good. Hi, Dennis. Hi, Abyss. Thank you for coming.
Dennis.
Hey all.
Hello,
Yo.
Billy Dennis.
Hi,
How are you,
everyone.
man? Hello, hello, hello.
I did some of the reading beforehand. This should be a very interesting conversation.
It should be interesting enough to keep Jamie awake even though it's 2 a.m.
I never get tired listening. In fact, I always come away from these being pumped, like weirdly pumped. Like, yeah, let's do science!
Yeah, agree.
Yeah, science!
Yeah, let's do science. It's a verb now.
That's our motto from now on.
Let's do science. Yeah. Was it? I saw there was a room there called "Do Atheists Logic Well," and they were talking about atheism and stuff in there, eh? And I popped in and just popped kind of out. They were having their own kind of debates back and forward, but I just love the tongue in cheekness of it. Do they logic well?
Very nice. I just quoted you in the chat, Jamie. I hope it's okay with you.
That's
I wrote your
okay.
motto in there.
Welcome, Dr. Shah. Looking forward to your questions and input.
Hi everyone. It just started, right?
We're just about to get started.
We're awaiting our guest.
Yeah, I wrote emails with him today, so maybe, I don't know, maybe he has issues coming. I just wrote him an email to
He'll
ask
be
if
here.
everything is
He's on his way. Hey, Serena. He's finding parking or something.
Hello.
exactly.
Hi, Serena.
Hi.
How's your day?
Oh, are we getting started on that?
Hey, Twan.
Hello.
Twan. Welcome, guests in the audience. Feel free to ping your friends. We've got lots of room. We'll get started in a little bit. We hope.
I was thinking of looking up specifically what the doctor was going to talk about today, but I like to be surprised just now, so I don't know where this is going to go, but I'm looking forward to it.
Okay, I won't tell you.
Spoilers.
Yeah, let's see. And I can make like general announcements. Maybe I should share the Discord.
You have a
Nice.
I heard you mention that you've got a
Yeah,
Discord thing for this.
let me get the link. Let me find it.
Well, where's Dr. Lent?
We are waiting.
Oh,
So
okay.
in the meantime, we can have some fun in general.
We were hoping you
the
could tell us
notepad.
that.
Exactly. So
There he
we.
is. Welcome, Nathan.
Oh, okay.
Hey,
Then I'll share it later.
welcome.
Okay.
Hi.
Hello, welcome.
Thank you for coming.
Can you hear me?
I hope you
Yeah.
found a good
Okay.
parking spot. I'm sorry, that was Victoria's joke earlier.
Oh, what did I say?
The
You
parking.
Hey,
can use it though.
parking is a
Oh yes,
So
go for it.
I asked if you.
parking is a nightmare in New York.
Do you need a pass? Do
Yeah.
you need like?
Yeah, thank you so much, Nathan, for coming. And I'll introduce you, and then if that's okay with you, Victoria asks like a question or two so the audience gets to know you a little bit better as a scientist, and then the stage is yours to introduce your research.
Okay, that sounds great.
Perfect. So welcome everyone to the Science Society today. We have a really great guest speaker here today. Meet Dr. Nathan Lents, and I will tell you a little bit about him. I'm sorry, my I'm sorry, my phone is bugging out. Yeah. Okay, so he's at the Department of Science in John Jay College, the City University of New York, and he got his bachelor in biology at the St. Louis University and his PhD in pharmacology and physiology at St. Louis University School of Medicine. And he did his postdoc in genomics at the New York University. And he has now his own lab, and his lab has two active projects. First, he's studying the recent evolution of the human genome in an effort to help to understand the genetic underpinnings of human uniqueness. The lab is engaged in a genome scale effort to discover and characterize these human-specific genes. And in general, the goal is to develop a DNA coding system for species identification of trace plant residue recovered from crime scenes, and the development of microsatellite markers for forensic DNA fingerprinting of plants, two tools that are currently lacking in the forensic toolkits. He also wrote popular science books and articles, and he has a blog at the Human Evolution blog, and he writes for also Psychology Today. Under the tagline "Beastly Behavior: How Evolution Shaped Our Minds and Bodies." Yeah, so thank you so much for coming. We are very honored to having you here today. And Victoria, go ahead.
Thank you, Katarina. Sorry, we're Nathan. We are
Yes.
so excited to have you here. While we were waiting, we were just lightly discussing that. So, if you don't mind, I'm wondering a few things, general things, and I'll ask you a question, and please feel free to answer it as narrowly or as broadly as you like. I'm interested to learn about how people arrive at their field of study, but backing up a little bit from that, how you decided or how you first became interested in science or knew that science was something that you wanted to spend your time researching or just being involved in.
Okay. Wow. Well, that's interesting line of questions. Let me. So I'll start with what you asked second because it leads into what I'm doing now. I I always liked science as a kid. Just good at math, good at science, kind of science nerd. As a kid, I liked science fiction and all that kind of thing. But at the time and place I grew up, when you express, I didn't know any scientists. Nobody I knew any scientists. So when you express a affection for science, they think, oh well, you should be a doctor. that's, you know, a medical doctor. That's all people think of when they think of as science. You know, like I said, at the time and place I grew up. Well, so I kind of marched through thinking I wanted to be a doctor, but I got to college and they made us do an internship in the operating room and an internship in the emergency room. Or I think you had some choices actually, but those are the two I chose. Well, I mean, those did not go well. I mean, it was immediately apparent that I not only was not cut out for medicine, but also not interested. I found it incredibly boring. I remember being in the locker room after an operation. I watched a 4-hour operation with another intern, and we just sat there and watched the operation. I remember thinking it was the longest 4 hours of my life. It was just incredibly boring, just watching paint dry. And even the surgeons were really nice. They were trying to explain everything to us, but I just did not find it interesting. And then we were in the locker room, and my fellow intern, just as I was about to open my mouth to say that, says, "Wasn't that great?" So I knew we were just different there because he was having the time of his life, and I couldn't have been more bored. Anyway, so that's when I was like, what else can you do with a science degree? Oh, I know, be a scientist. Because really what interested me is how everything works. You know, I wanted to take things apart and understand how they work. And I wasn't comfortable around people who were ill. So to science I went. And I've always enjoyed biology a little bit more than chemistry, although I do like chemistry. And my very first research project involved working with DNA. I was trying to get DNA into soil bacterium, a gram-positive soil bacterium. And then my second project, I was looking for genes responsible for resistance to nematodes in soybean plants. So these are industrial, kind of projects. And long story short, every project I've ever dealt with, I've been dealing with DNA, particularly DNA sequences and how DNA sequences work. So if you look at the different areas that I've published in, things I've written about, it looks like very eclectic, or some might even say unfocused, my research interest. But actually the common thread in all of them is I want to know how DNA sequences work, how they actually code for this incredible, thing that we call a human being. Not that all the work is done by DNA, but certainly most of it is. Most of the information is there. So I've still, to this day, am fascinated by how DNA sequences actually code for things. So that's what drives me. That's the interest that has pushed me through my career. Another thing I might say is that there's many forks in the road as you're pursuing a college, grad school, postdoc, and then whatever job you take after that. And my life could have turned out very differently at any of those choices, but I was really drawn to college life and an undergraduate specifically. So even though I did all my training at medical centers, I was very happy to get away from medical centers when it came time to set up my career, because I just love being in college, being around college. It's sort of that atmosphere of intellectual life and young people with young ideas. And I mean, everything is at the forefront of our culture is being discussed at the university. So I never wanted to leave.
Thank you for that journey, that in-depth journey along your thought process. I have to say, I think it would be presumptuous to say to give a judgment and say that somebody's research looks, what did you say, unfocused, because I don't know exactly what the path was that led you to each research project. So I wouldn't presume that. I'm just thinking maybe people who are working in science maybe would be able to recognize that something that might look like chaos is not at all.
Yeah.
But I also find that it's interesting, your book, your tagline, "Beastly Behavior: How Evolution Shaped Our Minds and Bodies," because you are really sharing exactly, it sounds like you're very in touch with your own behavior and what drives you because of what you said about observing the operation, the surgery, and then where you are now, and what you're doing, the evolution of the human genome, and just how your description to understanding the genetic underpinnings of human uniqueness. So yeah, thank you. And then you've explained how you arrived exactly where you are today. So thank you so much. And the floor is yours to
Okay, great.
hear
Well,
about your
Since you are interested on the more personal side of things, I can actually tell how this project actually started, rather than what I would say to just to a pure science talk.
Thank you. I
Because
just want to interrupt you just for one moment because I see you have the little party hat on, which indicates that you haven't been here for longer than 7 days. And so I
okay,
want you to know that when you see our mics flashing like this, can we flash our mic?
oh,
It means that we're applauding. We're silently saying "yay" for whatever was just said.
oh,
So
okay,
I was applauding you, recognizing that you may not realize it. So now you can know. So all right,
okay.
now Nathan, the floor is yours.
Okay. While I was a faculty member at John Jay College, I was increasingly taking on administrative roles. And so I was getting further and further away from my research lab, further and further away from even teaching, and doing, part-time administrative roles. And eventually I got to a full-time administrative role as the director of the honors programs. And I did that for 4 years. And for a number of reasons, I was very eager to step down. And I realized I was just too young to leave science behind, leave my research lab behind. It's just, I don't mind, if we end up there eventually, but it was just too early. At that point, I wasn't even 40 years old. So I went back to the lab, but the problem is if you haven't been running a research lab for 4 years, all your reagents are expired, all your, you have to rebuy everything. And I didn't have a steady stream of projects that were keeping the funding streams coming and all of that. So I decided to go back into what I did for a postdoc. About half of my postdoc was in computational biology. So I said, well, that stuff, I have what I need, and I can write grants for better computers, for a fraction of the cost of doing bench work. So I said, let me go back into computational biology. And my real interest is, as I said, is in DNA and how it works anyway. And I've been interested in human evolution because all of the work I've been doing writing on human evolution. So I decided, why don't I sort of put these interests together, human evolution, my passion for genetics and DNA, and then also this desire to get back into computational biology. So I put all those interests together and said, well, what's the big unresolved question in human evolution? And I mean, there are many unresolved questions, but the biggest one, as I looked at it, was what is the genetics of human evolution? We know a lot about our anatomy, our morphology, and sort of the patterns and the selective forces that that led us to evolve the way we did. But we have almost no understanding of the genes that are underneath all of that. And it's not, genetics isn't everything, but it's going to be certainly most of it when it comes to the anatomical changes associated with how we became so different. In just less than 7 million years, we really transformed. From just another run-of-the-mill African ape into whatever it is we are now. So I wanted to take a look at the human genome and just sort of find areas in it that were either underexplored or underappreciated. So what we did was a very simple approach, and this is figure 1 of the paper. We just lined up the human chromosomes with their chimpanzee counterpart. They're our closest relative to chimpanzees, and they've been evolving too. So it's not just they're not ancestors, of course, but we can sort of deduce the ancestral state of some of these chromosomes by comparisons with the other with our fellow African apes. So I started with the chimpanzee chromosomes, and what we realized, and our goal was to just find what we call deserts of conservation, or you could say islands of divergence, meaning parts of the chromosomes that just really look different between the humans and the chimpanzees. Very naive question. This is a very naive thing to do, by the way, because people have been working in this field for some time. But this is always the benefit of being a properly trained outsider, is you can come in with a very fresh look and ask naive questions, and sometimes you find things that other people haven't overlooked because they've been steeped in, this is how we've always done it, or this is what we've always understood. So we just lined up the chromosomes, and our goal was to find these little deserts where there was marked divergence between humans and chimpanzees. And my goal was to basically go through all the chromosomes one at a time. So we started with the smallest one, which is chromosome 21. The chromosomes are numbered in order of size, with some exceptions at the smallest ones when they were numbered before we actually had the size accurate. So even though chromosome 22 you would expect to be the smallest one, it's actually chromosome 21. So we started with chromosome 21, and we lined it up, and it's an acrocentric chromosome, so we knew there'd be differences between the short arm and the long arm of the chromosome. But we lined it up, and sure enough, it just didn't line up very well on the western end of the chromosome, west of the centromere. So this is the short arm. So the short arm of the chromosome just really didn't line up very well, but the long arm of the chromosome lined up perfectly well. So we noticed that the short arm had a lot of divergence between how the chimpanzee chromosome looked and how ours did. So we started then saying, well, are humans the weird one, or are chimpanzees the weird one, or is this just this chromosome is a mess in all the apes? So we did the line with other apes, and so we did pairwise, basically all the combinations, because we have the ape genomes that we have access to, that everyone has access to, besides humans, are the gorillas, the orangutans, and both species of chimpanzees, the chimpanzees and the bonobos. So we lined them up in all of these pairwise combinations. What we noticed is that all of the other apes, their chromosome 21 lines up really well throughout the entire length of the chromosome, short arm, long arm, centromere, everything. But really, if you line any of them up, and this is figure 1D in the paper, if you line any of the apes up with their chromosomes, with our chromosome 21, and you'll notice that the short arm just does not align very well at all. So there's a lot of sort of unique human sequence there. But the other apes all line up to each other fine. So it really is human are the outlier. So we decided to zoom in, and basically just did this analysis with more and more precision as we zoomed in on these specific divergences, because you can do this. You can do the settings of these alignments almost any way you want them. You can have minimum length. You can have minimum sequence similarity. There's lots of ways to do this, and you can be tolerant of either strand or both strands. It's lots of dials and knobs that you can twist on these alignments to get more fine-tuned results. But you don't want to do it with the entire chromosome because it's many megabases of sequence. So we wanted to keep zooming in. Is basically what we did. And figure 2 of the paper shows the result of that zoom in. We really kind of narrow things down to about a 1 megabase region that has huge chunks that are just not conserved at all between the human and the chimpanzee, and even individual stretches, tens of thousands of base pairs where there's simply no sequence similarity anywhere in the chimpanzee genome to what the humans have. So this was a very unexpected result. We didn't expect to have these huge chunks of unique human DNA sequence. Now, the next thing we did was we just asked, well, what is there? What's going on in this roughly 1 megabase region that we were focusing on? Well, actually, I'm sorry, the window of 1 megabase actually was set by the next result. So I shouldn't have said that too early. We were looking at a larger chunk, more like two megabases actually, but then we zoomed in on 1 megabase once we saw what was there. So figure 3 shows what we found, and not what we found, sorry, this is what is the annotation of this region of chromosome 21 that's out there. And what we noticed is actually 3 families of microRNA genes. And these microRNA genes they have a gene named MIR and then a number. So these are all the MIR genes. We have about 1,600 of these genes in the human genome, but we noticed these clusters. And what was interesting about these clusters of MIR genes were that they were in these repeating patterns. So we had 4 members of one family, 2 members of another, and 2 members of a third family. But they were repeated in ways that sort of looked like tandem repeats, but not quite. And then another repeat looked like an inverted repeat, but not quite. We just saw like whole chunks of this chromosome seem to have been duplicated in a weird way. What we also noticed is that in this region, they're not just these microRNA genes, there are also arrays of ribosomal RNA genes. So the way that ribosomal RNA works is this is transcribed and then chopped into smaller pieces. So you have a sort of mega ribosomal RNA that then gets processed into several parts. And then those parts are what are actually used to build the ribosome, the RNA component of the ribosome. I don't know if you know, but the ribosome is more than 60% RNA in terms of mass. Anyway, so within these RNA genes seem to be the MIR genes, the microRNA genes, sort of genes within genes, which is possible with RNAs, of course, because there's not a reading frame that has to be maintained. So, and also shown in figure 3 at the bottom, we just show that these regions are enriched for CPG islands all throughout the annotated gene regions, indicating it's been maintained by selection, probably transcriptionally active. Actually, it doesn't show that it's been maintained by selection. Actually, it shows that it's likely to be transcriptionally active, and therefore it would be maintained by selection. That's a different test we would do. Okay, and then as we zoom in smaller and smaller, what we were able to do is what's called self-synteny, where you compare sequence to itself. And what we found was there really are these large chunks of what we call segmental duplications. They're of various sizes, and even chunks within chunks of DNA were subjected to this segmental duplication. Now, it should be said that segmental duplication itself is not terribly unusual. When you have large stretches of repetitive DNA, these do tend to expand over time. They can also contract, but generally we don't see the contraction events as often as we see the duplication events. This was driven by basically misalignment during meiotic recombination. So some DNA gets pulled from one homolog to the other during meiosis, during crossing over. So we found evidence that these chunks within chunks within chunks of repeated duplications have occurred. But interestingly, the model that we proposed is that within the process of those duplications, some regions that code for ribosomal RNA were sort of born into microRNAs, because if you take a chunk of several different regions of ribosomal RNA, it's very easy to get a hairpin structure that looks a lot like a microRNA. So if this chunk would be created by duplication, such that it created a hairpin, that hairpin could be active in the microRNA system of gene expression. Now, I haven't told you what microRNAs are, and that's where things start to get interesting. So microRNAs, if you don't know, are one of the newest classes of molecules ever discovered. They were discovered really in terms of what their function is less than 20 years ago. And these microRNAs target genes for post-transcriptional silencing. So it's a way to turn a gene off, a separate gene for some protein, to turn it off even after that gene is transcribed into messenger RNA. You can then target that messenger RNA for destruction. So it's kind of an additional level of gene expression control, these microRNAs. So, but they're very small, very compact, tiny little molecules, basically just a hairpin. The overall hairpin can be as small as 60 or 70 base pairs. Most of them are in the 100 to 200 range, very small genes, and but they can have far-reaching effects because a single microRNA can target 300-400 different genes for silencing. And it's important to remember that silencing isn't always, in fact, isn't usually complete. It's more of a turning the volume down rather than turning the power off. But still, it would affect gene expression. So a single microRNA can affect hundreds of targets and being expressed all throughout the body, or in just one tissue. Both extremes are possible. There are microRNAs that have 7 targets. There are microRNAs that have 700 targets. And there are microRNAs that are expressed in every single cell of your body, and there are microRNAs that are expressed only in neurons, for example. In fact, there are many of those. So all things are possible with microRNAs is kind of the way to look at it. Is they're really capable of very exquisite, fine-tuned control of gene expression, but it could, with far-reaching effects. So our hypothesis at this point is summed up this way. Chunks of segmental duplications over evolutionary time occurring on chromosome 21 created new microRNA genes out of ribosomal RNA genes pre-existing. And so the creation of these microRNAs were then active, and actually I'll get to that in a second. Some of these microRNAs anyway were active in silencing genes and therefore affecting the organism. And once you start having some kind of effect, a phenotype in genetic terms, then those microRNAs would be subject to natural selection. And over the time were refined into what we now know as MIR 6724 and MIR 3648. These are the two of the 3 families. The third family member we don't believe is active as a gene, but the other 2 we know are. We know they're active because they've been detected in screens. They've been studied in several tissues. They are even found in cerebrospinal fluid in the case of MIR 6724. So the point is, these are human unique. No orthologs exist in chimpanzees or any other ape. They do seem to encode for microRNAs. And what we were able to do was predict what the gene targets are of these microRNAs. So these are human unique microRNAs. By the way, we did find them in the ancient genomes that are available, Neanderthal and Denisovan genomes, for those who are interested in those, and in identical or nearly identical form. But in any event, we were able to predict what the targets of these new human unique genes are. These are very young genes. And actually, they show the signs of being young, meaning they have diverged, they have much lower sequence conservation among the apes, and even among human population, there's some variations. These are very young genes. What we found when we predicted the targets of these microRNAs, so there's tools you can use. Actually, we wrote our own algorithm for this. to actually detect what the targets of these microRNAs are, because the microRNAs target the genes that they do by simple sequence matching. Now, there's some variations on this theme. Not all hits are identical, and there's only a certain 7 or 8 base pair region that does the targeting, and the target has to have that sequence in its 3' UTR. Number of conditions to be met, but the point is that we can actually run this algorithm and detect with fair amount of accuracy what the targets of the microRNA are. Now, there's no substitute for validation of those targets, so that would be future studies. But using just the predictive software of what these microRNAs actually target, we were able to look at the lists of genes that are affected. And it is quite remarkable what those genes are. So there's a program or an algorithm called gene ontology, which actually looks for overrepresentation of certain of whatever category of genes. So if you have a list of genes, let's say you input 100 random genes into the algorithm. First of all, that would if you pick 100 at random, nothing would happen because it would say there's no enrichment. But what it would do if these were a meaningful set of 100 genes, it would say, wow, these are enriched for genes involved in, I don't know, skeletal muscle function, or these 100 genes are enriched for those involved in the cell cycle and growth control, and so on. So all genes in the human genome, well, 20,000 of them anyway, have been characterized and put into different categories, and these are hierarchical categories. So what we did was we generated target lists for our human unique microRNAs, and then went and looked at what kinds of genes are in that list of their targets. And what we found is there's a really remarkable enrichment for genes involved in development of the nervous system, specifically development of embryonic development, regulation of neurons, regulation of synaptic formation, several of the categories that basically that what you would expect to find if you're looking at genes involved in the development of the human brain. And so, and remember, there's no prior knowledge that we were interested in human evolution that goes into any of this, right? We were just feeding lists of genes into a program that's designed to detect enrichment for categories of genes, and they found enrichment in our lists. So we were obviously very excited about this, and we're following up on this with several related projects because what we really conclude from this is that microRNAs, and I don't just mean these two, we focused on these two, but microRNAs in general might be a very underappreciated engine of human evolution because they can do a lot with a simple one base pair change. You can affect gene expression of hundreds of genes in various tissues of your body. So it seems to me like they would be sort of good fodder for unique evolutionary events in our lineage. I am basically right at 9:30. How did I do in sort of summarizing that on time?
You can go on as long as you like. I was curious. How long we have the honor of your presence.
Well, I mean, I'm fine for a little while. I'm not really going anywhere. If you want to ask some questions, I think that'd be great, and then I can, see what else I want to say after based on the questions.
We're
I'll
really
chime in
good at
with one.
keeping reasers up all night long, so feel free to set a firm boundary with this group.
Okay,
All right.
well, I have to tuck my son in at some point, so that'll be the hard cutoff.
So I have a question. So fascinating story and fascinating results. And, so am I to draw the implication for the distinction of the human brain and its development relative to close neighbor apes is that in essence the microRNAs silenced or reduced the regulation of the previous architecture in the sense so that it allowed an expansion of the neocortex, for example, or other types of neural tissues.
Right. So I would say that that's a reasonable hypothesis to make. Our data don't say anything that precise. I wish we could say that. But what this does do is sort of point us towards some genes and some tissues at which these microRNAs might have been working in. We do know that compared to chimpanzees, the human brain is much expanded neocortex, as you mentioned. We also have much more specialization throughout the cerebrum, lateralization, and there's quite a bit of developmental change in the human cerebrum. So, these microRNAs, we're certainly not working alone. I don't mean to say that we've solved human evolution here, but I think added in with some of the changes to protein and other regulatory reasons for all these genes, I certainly do see, or at least the possibility of a role for if not these two microRNAs, other microRNAs in this process.
Okay, who else has questions? I know we have some flash mics. Okay, Abyss, go for it.
Hi, Nathan. Yep, a great presentation. I was listening very attentively, and I have a couple of questions. The first one being um, I guess it's based on what Serena was asking. I was when you mentioned something about particular these RNAs being particular to humans, I was also curious if they also play an active role in targeted cell senescence, so that for example when the neocortex is actually forming, if they actually can activate or deactivate cells so that it can have this, a gyrated bulbous structure that we have during development.
Yeah, so the short answer is yes, it's absolutely possible that programmed cell death could play a role in. Well, I think it's beyond question that programmed cell death plays a role in development of the nervous system. Whether these microRNAs are in some way connected to that, I don't know if we can say that yet. What I can say is that the genes involved in nervous system development that these microRNAs regulate, we didn't see enrichment for categories related to programmed cell death. But that doesn't necessarily mean anything, because the genes involved that we know are connected to programmed cell death, that's their job. That's their main sort of business. These microRNAs likely touch a earlier step in the signaling, if you know what I'm saying. So that's more of an output. Programmed cell death would be more of an output of some developmental process, whereas I would suspect that these microRNAs are working earlier in that process, earlier in the signaling cascade, in a way that's not necessarily connected directly to programmed cell death. It's just several steps upstream, is another way to say it. But that's what I found. Oops, that's just a guess, of course.
Gotcha. Thanks for that. My second question is there a plan to essentially activate those microRNAs in rodent models or non-human primates? I guess non-human primates would actually come later in the echelon of the research, but I'm curious if it's possible to sort of study particularly if these microRNAs play an active role in neural development in non-human models, if that makes sense.
Yeah, I mean, it's definitely an experiment, that like a conceivable experiment to get some kind of answers. Do we have plans? No, I do not have those plans, and I don't know if anybody else does. But an experiment where you would sort of try to provide this human unique microRNA into a non-human, there's some problems with that. It's not as clean as experiment as it might sound like. First of all, you have to remember that many other things were changing at the same time, some of which we know, most of which we don't. So it's really just a totally different organism. Another thing to keep in mind, and this is actually related to some of the work we're doing with the Neanderthal genome right now, is that you can't predict the targets of one microRNA in another species, because remember that they target a specific sequence in the messenger RNA of their target. It's in the 3' UTR. Well, that's been evolving also. So it would have a different target list in a different species. Now, there may be overlap, but that's even more reason why just providing a microRNA from a different species wouldn't be sensical in another species. It would just have a different set of targets. Yes, there might be some overlap, but I think you're just almost certainly to get an uninterpretable result, is the way I would put it. It would probably screw things up, but that doesn't really tell you what the gene does in humans. I mean, and the experiments that you could do to do that, of course, are not ethical. So what you would have to do instead is just sort of be able to visualize what these microRNAs are doing during human development. That's obviously not a simple task, but there are experiments that look at that, and we might be able to mirror some of that in other species. But it is a difficult question to study a human unique gene, because by definition, putting it into another organism really isn't possible. I mean, you can do it, but it won't give you a clean result. And of course, we don't experiment on humans. So, yeah, you have much fewer options, many fewer options. But these are
Yeah,
good questions. These are good
I think
questions.
that's the I think that's the risk with genetics, that often you ask, well, what if we put this into that? And then you have to work through it and realize, well, that's not actually ethical, because there's a creature, there's an entity, there's a being, so you have to find more ethical ways of navigating that. Unless, of course, you find yourself in a foreign nation state of some sort, like where the boundaries are not so well defined.
Right. I don't. I don't presume much of that's going on with human evolution so much, but you
Well,
never know.
China did that experiment on those two embryos, right? So that's what I'm kind of alluding to.
There's been a three parent baby that was born in China within the last 12 months, is what I was reading.
I think the doctor just got released from prison
Yeah,
too.
it's funny. You get funded by the government, and then they send you to prison, and then you're out. So it's kind of a weird trip that he took. But sorry, my apologies for disrupting your flow.
Dr. Shaw, I saw
Well,
you on
work
mute.
in the Neanderthal genome, and believe it or not, I have had someone email me one time to ask if they thought, I don't know who this person was, to ask if I thought we would be de-extincting the Neanderthals. Um, I, for the record, I do not think we will be doing that, nor will I. I certainly wouldn't advocate for it, but I don't. We're nowhere near to where that could be possible.
That's an awesome email, by the way.
Yeah, when you work on human evolution, you get weird emails.
Oh
That's
my,
very funny.
we could do like 10, I'm sure we could do like 5 minutes on like the weirdest emails you got.
Yeah,
Yes, please.
let's
Okay.
shout it
That's
out,
great
Can I ask a
direction.
question real quick?
please.
Thanks for that.
Dr. Shaw
So,
was
I think Dr.
first,
Shah
Kirko,
was
and then we'll
on
get
miking.
to
Yeah.
you.
First of all, thank you so much, Nathan, for sharing your, study with us. And for me, that was very interesting from the point of, mirror ribosome, microRNA that you just mentioned about it, because there's a use of them in cancer susceptibility for the candidate 7. If you just heard about that, also lots of use in drug development, because right now we have lots of development on microRNA base, and also for the checkpoint in the cancer therapy and immunotherapy. If you are aware of, also in another part, I just noticed that they have a use of synthetic CRISPR products of using this mirror kind of microRNA. That was very fascinating. Thank you so much for sharing that.
Oh, you're welcome. I do think we are just at the beginning of appreciating all that microRNAs do for us, or do to us, even if you want to think of it that way. So I think you'll be hearing a lot of exciting stuff in the world of microRNAs, not just for human evolution, but throughout biomedical science. I think it's underappreciated. I think we're just really getting our hands around it now.
I see a lot of potential in that for not gene therapy, because that would be something different, but for medical treatments, therapeutics.
Yeah, I do too. I think, especially because you can provide them in a way that's very specific to certain tissues, because that's often a problem with any gene therapy or genetic based treatment, is that you got to get it to the right place, and only the right place. A gene in one tissue is important, and a gene in another tissue can completely screw it up. Cystic fibrosis gene is the CFTR transmembrane regulator is a great example of this, because it's very important in the tissues that it's important in, and it will completely kill many other tissues as well, because of it totally throws off their ion balance. It's a chloride channel, and without proper control, you can't just leave chloride channels open. So that's why the gene therapy is so tricky, is that you could kill with your cure. So microRNAs, the advantage of those is that these nice small compact genes are easy to deliver, and you can just simply put a tissue specific promoter, something that is not which isn't possible with big protein coding genes. First of all, you can't, the promoters are different anyway. I didn't mention that, but yeah, they make a good delivery system, and you can make them permanent, you could make them transient. It's really, I think it's underappreciated. So we'll see where it goes. But I'm not, just for the record, I'm not in clinical research right now. I'm not doing anything related to that. I'm studying them in human evolution.
Would you class that under off-target effects?
Well, I mean, I think that the term off-target effects is a pretty specific term in pharmacology, so I don't believe it's used in this context, in the context of gene therapy or genetic treatments. So no, I wouldn't, because I don't think it's the
Well, I
right.
guess it's so. For example, the different protein or slightly different expression or regulation could have drastically larger effects than is expected. Is that a possibility?
Oh yeah, absolutely. I mean, that's why genetic treatments are really, you have to be very careful. The potential for danger is great. Yeah.
I guess that's what the
Can I
question
ask a
meant.
So we
question?
had
Yeah.
Kirko, then Jamie and Tuan on stack for questions. So Kirko.
Thank you. I was starting to forget. It's a quick question, possibly. But I was kind of thinking about. I kind of came in the room late, so I don't know exactly all you talked about beforehand. But I just think microRNA is like really cool, and I think sometimes it can be used to like kind of turn up and turn off like gene expression. And like I recently watched a video on like how like whales uh, really don't get cancer, and what they were attributing it to was like the increase of the amount of like tumor suppression genes. And I was thinking like one way that like genes can kind of like uh, increase in number diversify is like through transposons. So I was kind of wondering, or transposable elements. So I was kind of wondering if there's any like correlations with like certain micro mRNAs and like transposable elements where like maybe like certain genes that like have times just happen to have like a similar or the same microRNA. You know what I'm saying? Because I feel like that may shed some light into like this whole human evolution type of
So Kirko, it's really interesting that you put these together, because yes, absolutely. First of all, there is a connection between transposable elements and microRNA gene creation. That's not what we found in this region, but we spent more than 6 months chasing that hypothesis. So I want you to know that was a very good suggestion that you came up with, because we thought the same thing. That and indeed there are microRNAs that seem to have been carried along for the ride with transposable elements, which means they were copied into multiple family members. In fact, there's one of them, there's at least 9 family members of this gene that have been copied through transposable elements. Generally, what's interesting is they just create more of the exact same microRNA, and meaning that the genes might diverge a little bit, but they produce the same product, the same microRNA, because there's a lot of processing of the original transcript. So the final thing is only like 21 to 22 base pairs long. But it's a duplex, so and both sequences can target. But anyway, the point is that transposable elements are a vehicle for gene duplication, including of microRNAs, and that's why RNA genes are so much first of all, harder to study than protein coding genes. I mean, the annotation of the human genome. People talk about all the areas that we're just now learning to annotate. Those are all RNA genes. We figured out how to find protein coding genes a long time ago, because protein coding genes tend to follow a lot of the basic rules the same. You find a CPG island, you find a Tata box and a cat box in close proximity. You find underrepresented codons. You've got 6 reading frames. So if there's a start and a stop within this, there's all these rules for finding protein coding genes. It's not that hard. But RNA genes, there's no rules. It's really hard to find RNA genes, with nothing but sequence. So what you have to do is get better and better at detecting transcripts, and then finding the genes, based on complementarity to the transcript.
But the problem with that, I don't know if you guys know this, if you've heard of this, but the ENCODE project has taught us, which is that there's a lot of spurious transcription. There's a lot of just kind of junk RNA, meaning junk transcripts. There's sort of a background rate of transcription of just anything throughout the genome that is meaningless and functionless, most of which. But gosh, what a reservoir of molecules for evolution to tinker around with. They're sort of there in the background, waiting to be used. But anyway, and so it's really hard to find RNA genes in the human genome, just in general, because they don't follow any of the same rules that coding regions do, first of all. And secondly, there's transcription everywhere, pretty much throughout the chromosomes, and at least low levels of it, unless you're really like, in an alpha satellite or whatever, but, heavily methylated DNA. But the point, how do we get onto this? Sorry, this happens to me. The transposable elements and human evolution. So that was our first suspect. But what we found, well, first of all, we did find transposable elements in the region, but they weren't associated with the microRNAs. So they were active. But the gene duplications we saw, I shouldn't say gene duplications, the segmental duplications that we saw were too large. Most of them, not all of them, most of them were too large to be transposable elements, first of all. So in addition to looking for those, we also were looking for other mechanisms. And of course, the logical choice is meiotic recombination, because when you have crossover events, I don't know if you guys remember your intro bio, but when you have a holiday junction, it's actually two junctions, right? And with a heteroduplex in the middle. And if you have misalignment, you can actually pull DNA from one homolog to the other. And then when the crossover is cut and resolved, you've actually deleted DNA from one homolog and increased it or duplicated it in the other. Now it depends on which one. They end up in different gametes and blah blah blah. So you should see insertions and deletions, or I should say duplications and deletions in equal numbers, but you don't usually. You find usually the expanded set, not the deleted set, for reasons we can talk about later. But the point, though, is that that's what we think we found. Oh, and how do we know that? Well, besides just the size, we also can look for anywhere in the genome. You can look for what are called recombination hotspots, and so there's certain sequences that are more likely to lead to recombination or have a recombination event. But remember that these would just show sort of yes, it is a sequence search, but it doesn't mean that that's exactly where the branch point happens, because remember that they slide. The heteroduplex slides. Well, actually, the holiday junction slides, and so it might begin one place, but it's somewhere far away by the time it's resolved. So what these hotspots tell you is that somewhere in that neighborhood, you're more likely to get a crossover event, which doesn't prove that that's how these happen by any means, but it's just sort of suspicious. And also the size is suspicious. And also there's only so many mechanisms, at least that we know of, that lead to these segmental duplications. So that's our working hypothesis. And how could we prove it? Well, we're going to get, hopefully over the next 10, 20 years, we're going to get access to more and more ancient genomes. We have genomes now that are 50,000 years old, 100,000, well, not quite 100,000. But I suspect if we start getting DNA from the fossils in Spain, the Cimo de los Huesos, from those fossils, those are quite a bit older than anything we have right now. So we're getting closer to, maybe not the midpoint, but further and further back in time if we assume that these, they may not be our ancestors, they're going to be a sister branch, though, so we can sort of deduce the ancestral sequence from that. The older we get, the closer we'll get to whatever that ancestral sequence was. And that might help resolve some of this as well. We won't be able to see all of these duplications in action, so to speak, but we can see some of them, that have happened in the last, hopefully, 100, 200,000 years. We'll catch some of the other ones. And then we'll know. Well, we won't know, but we'll have more data. I think I could probably do one more question, and then I might have to bail.
I was going to, can I ask you, Doctor, you were mentioning about the microRNA and how there could be a lot of potential in it by making it like, tone down things and tone up things. So if I'm understanding it correctly, like being able to manipulate the DNA and everything from a kind of, 2 steps away, like a control panel in the computer or something like that. But did you find any patterns to how micro RNA actually behaved? Like, because you said it can affect little parts in small ways or a lot of parts in big ways. Did you find any
Um,
behavioral?
yeah, so we haven't done that yet. We haven't started to try to observe the function of these in vivo or even in vitro, actually. But other researchers have previously published about these microRNAs in terms of what they do. There wasn't any evolutionary look at it or even a look at the gene itself. But the both of the microRNAs that we were interested in that are human specific, and by the way, there are others human specific ones that we just haven't published those yet. Um, the human specific ones, these two, have been detected, and they are expressed in the brain. Both of them are in the adult brain. Both of them, one of them is actually detected in cerebrospinal fluid. They've both been associated with disease states in one way or another, because there's a lot of papers out there that just scan the whole global picture of microRNA gene expression in, say, ovarian cancer or skin cancer or whatever, or even just this tissue or that tissue. So while this doesn't really talk about their function, what it does do is show that these are active genes that create a gene product, that's detectable. And also both of them, in at least one paper each, have had some of their targets validated, meaning that these things really truly affect the expression of some of their gene targets. So we know that they're functional in the human body, and that no other creature except humans have them, and that our closest relatives, Neanderthals and Denisovans, also have them. Although we don't know for sure if they've been detected there, of course, but they had the gene. So,
And and
mhm.
if you didn't, and so if you haven't looked into, if you've not been able to look into yet any pattern there, I suppose then you wouldn't have any indication if particular instruction of microRNA was like hereditary. Like a family line would have an
Okay.
RNA code.
Yeah. So there are no clinical diseases or clinical anything actually associated with either of these genes. So there have been no. There's a database that's called ClinVar that any particular genetic modification that has been associated with a certain disease stage or clinical condition of some kind. Those have been detected. Of course, not everybody's looked at every single thing yet, of course, but there is a pretty big database of ClinVar, so genetic variation that's been known to associate with disease. Neither one of these have any entries in that database. That doesn't, it's evidence of absence is an absence of evidence. Or sorry, absence of evidence isn't evidence of absence. But by now, there's very little. Well, I should say there's no evidence yet to think that these play any role.
Thank you
So,
very much.
well, thank you all. It's been a great conversation. I need to say goodnight now, but I had a wonderful time having this conversation. I'll be listening in on future ones.
Thank
Thank
you
you
so
so
much,
Thank
much.
you.
Nathan.
Thank you.
Thank you,
Thank
Nathan.
you.
Okay,
Bye.
All right. Have
Thank
a
you
next
very
nice
much.
evening.
Take care.
Have a
check
good
for
evening.
forensic
Okay.
research. Okay,
Thank you so
bye.
much.
Thank you. Bye.
Thank you.
Yeah, thank you everyone for coming. Thanks for asking great questions and participating in the discussion. I really appreciate it. Thank you, Nathan, for coming. Really appreciate the time you took for doing this. And yeah, if you like this sort of rooms, follow the Science Society Club, and we have more rooms coming up. I don't know if anyone of the other moderators wants to say something before I make like the regular announcements.
I had a million questions that we didn't have time to get to. I wanted to know what it takes to the cost to run a sequencing lab, the implications of the recent announcement that the remainder of the entire human genome had been sequenced, and whether that was actually 100% true or not. From what I read, it seems to be true. I had so many questions.
Okay. Yeah, we can invite Nathan back always and then ask more questions. He has also more interesting research from his forensic lab related research. So I think I think forensics are really interesting, and so I always wanted to learn more about like a real forensic research lab, so it's pretty cool. Yeah, let's plan for that. For inviting Nathan back, and I think that's a good idea. Tomorrow we'll have at the same time Dr. Liang from a company, and she will talk about carbon negative production of acetone, isopropanol. It's more like efficiency, climate related room. Then we'll have Dr. Moraine. It's a more mental health related room regarding ADHD and hoarding. And then we will have on Friday, we still deciding, it will be more roundtable discussion type of room. We'll come with an interesting topic from recent news, science news, and discuss that. And then on Saturday at 1 p.m. EST, we have Dr. Marco Pizzini. He's a physicist from Italy. He's currently in France, and talking about his long distance electrodynamic intermolecular forces paper and his work. So, yeah. And next week, so we have a lot of more guest speakers. Please check out the calendar. Um, oh, right. If you would like to have a more detailed calendar that goes more into the future than what you can see on Clubhouse, we have a Discord channel, and we will start to use that more. Also, if you have like questions for us, suggestions, something that we should do or not doing yet, or just participate in the discussion, you can are welcome to sign up for our Discord server, and I'll put up a more detailed calendar on there for the future. I think we have booked until the last week of May. So great. Yeah. Thank you, everyone. If you have something to share still, please go ahead, and if not, we'll close the room for today.
Good evening, everybody. Have a good night.
Great.
Yeah, good night, everyone. Good night.
Thanks. Good night, everyone.
Thanks, everyone.
Bye,
Bye.
everyone.
Thank you. Good night, everyone.